Okay, so just to clarify about plasmids, on the AP exam or on the quiz we're going to take in my class next week, here's kind of the situation they could give you. They could explain to you what plasmid they were using. Remember, plasmid's a little piece of DNA from a bacteria, and it's being added to, in the cases, the samples I'm going to give you, it's going to be added to another bacteria. So they're going to give you a situation. They're going to tell you what the plasmid has, and then they're going to ask you, like which of these plates might it grow on? In which of these situations would it grow? So here's sample 1. The plasmid has ampicillin resistance. In other words, the plasmid contains, the DNA in the plasmid contains a gene that would make whoever picks up that plasmid resistant or protected from the antibiotic penicillin. So A would represent your answer if bacteria that received the plasmid would grow, and B, you would choose if you think bacteria that did not pick up the plasmid would grow, or your answer could be both, A and B, that you think both of them would grow, that they don't, whether they have the plasmid or not, or your answer could be none of them would grow. So let's look at our sample plates. So number 1 says LB agar. Now, LB, just a reminder, if they don't explain it to you, it stands for Luria broth. Luria broth agar is basically food. That's all it is. So it's not screening anything. It's just basically agar, which is like a seaweed gel. And added to it is LB, which is food. So on plate 1, what would we expect to grow? Your answer should be A and B, both. Why? Because you're not screening them in any way. If they picked up the plasmid, they'll grow. If they didn't pick up the plasmid, they'll still grow. Everything would be able to grow because you're just basically feeding them. Plate B. LB agar again, but this time we added, this should say ampicillin. So on this one, only A would grow. Why? Because the ones that did not receive the plasmid would be killed by the ampicillin. So we would only expect bacteria that picked up our plasmid to grow on this plate. We are screening them using the antibiotic. Plate 3, LB agar again, food, plus the antibiotic streptomycin. Your correct answer here, none of them would grow. Why? Because this antibiotic, we don't have anything listed here that helps them survive in the presence of this antibiotic. Therefore, this antibiotic would kill everything. All right, let's look at a second problem. This time, the plasmid causes the bacteria to turn blue in the presence of lactose. Keep in mind, lactose, you should know what that is. It's a disaccharide, so it's a sugar. It doesn't kill bacteria. Lactose would never kill a bacteria, but this plasmid would cause the bacteria to make a protein that turns them blue when this is present. So, LB agar, this is just food. We would expect all of our bacteria to grow, whether they picked up the plasmid or not. And now, LB agar plus the addition of lactose. Now, this one's kind of a trick question. Your answer is both of them would grow. Again, why? Because lactose doesn't kill bacteria. But what would be true, they would both grow, but the ones that received the plasmid would be blue. The ones that did not receive the plasmid would just be plain old white bacteria. So in this experiment, we are still screening them because we would physically be able to see the ones that picked up the plasmid on plate 2. Even though we wouldn't kill the other ones, we would be able to tell which was which because the ones that picked up the plasmid would turn blue due to the presence of lactose. And this is kind of like what we did in our lab as well. Because in our lab, if you recall, we had a plate that contained the sugar arabinose. That sugar was the trigger for the bacteria to make the glowing protein. So if we had had a plate that contained LB agar plus arabinose, everything would have grown on that plate. But the ones that had picked up our plasmid would have glowed. And the ones that did not pick up our plasmid would have just been white and they would not glow. Now, we didn't do a plate like this. If you recall, we did a plate that had LB arabinose, that's what ARA is, but it also contained ampicillin. Since our plate contained ampicillin and the bacteria that did not pick up the plasmid were killed by ampicillin, we did not see that result. What we saw on this particular plate was just glowing bacteria. They all glow. Why? Because only the ones that picked up the plasmid survived the ampicillin. But if we had had a plate that was LB plus arabinose, then that plate would have had all the bacteria growing, whether they picked up our plasmid or not, but the ones with the plasmid would have glowed, and the ones that did not pick up the plasmid would not have glowed. All right, last one. A plasmid that allows the digestion of lactose. Again, lactose is a sugar. It's a disaccharide. If a bacteria does not have the lac plasmid, it would not be able to digest it, meaning it just wouldn't be able to eat that particular food source. So plate 1, LB agar. What would grow on that plate? Everything, because again, we're not screening it. There's nothing on this plate that would prevent a bacteria from growing. We're feeding them food, so whether they got the plasmid or not, they would all grow. Second plate, LB agar with the addition of lactose. Now this is also kind of a trick question. The answer here is they would both grow. Why? Because as long as we're providing them with LB, luria broth, this is food. So the ones with no plasmid, F, they still have food to eat. They're not going to die. They'll just eat that. The ones that have the plasmid will additionally be able to eat this. But since we've provided a different food source, the LB, everything will grow. And then our final plate, lactose only agar, meaning sometimes they'll write this as minimal media, meaning you've given them basically the agar, the gel, but no food in it. So minimal media plus lactose. On this plate, only the plasmid ones would grow. Why? Because the other ones would starve to death. If the only food available is lactose and they can't digest it, they're going to starve. So this would be, again, another way of screening them. So these 3 samples are basically the 3 ways that we would screen our bacteria. All right, going back to the PowerPoint, quick little thing about electrophoresis. So we did a lab with this last week, and we even tested already on how to draw the bands in an electrophoresis. But one thing that I didn't talk to you about was how to recognize an electrophoresis, recognizing a child to a parent. So quick summary of electrophoresis. We cut up the DNA with, we need to use restriction enzymes. Restriction enzymes are going to cut everybody's DNA a little bit different. So we'll get a unique band pattern for different people. You put the DNA at the negative end. You should already have known that. We put it in a buffer and we plug it in to a power supply. When the current starts flowing, the DNA is going to be repelled by the negative side because the DNA is negative, and it's going to start moving towards the positive side. Smaller pieces are going to be able to get through the gelatin faster, or the agarose, and the bigger pieces are going to get stuck and they're going to travel more slowly. So we're going to get a unique band pattern, and we're going to have separate pieces. So this is just a picture of, there's a box here with the buffer solution. The gel is here, the power supply is here. You won't be tested on the parts of the supplies. So this is a sample of an electrophoresis. So this DNA sequence happens to be 50 base pairs long. Now I already showed you guys how to cut this with restriction enzymes. Think of this as one strand of DNA. So in other words, these 2 sides are connected. It's just the 5' to 3' and the 3' to 5'. This is 3', this is 5'. So this is one strand of DNA, the same as if we had drawn it vertically and drawn, A to T, G to C, like this. So if we were to cut this, I'll just give you one example. Let's say we cut it with this enzyme, HEY3. If you recall from my previous lecture, this one cuts when it sees CCGG. So if we go over here to the left, here's CCGG, and it cuts right here in the middle. And then on the other side, reading 5'3", here's CCGG, and it cuts in the middle. This would represent one cut through the whole DNA. If I was to run an electrophoresis now of this DNA, you could be asked, how many pieces would you get? Well, you would get 2. This would be one piece, and this would be the second piece. And where would you see the bands? Well, if I literally count 1, 2, 3, 4, 5, my original piece was 50 base pairs long. This is now 5 base pairs long, which means this is 45 base pairs long. So I would expect, if I was to cut it with this enzyme, that I could make a scale over here, 50, 40, 30, 20, 10. I would expect to get one band way down here at 5, and one band way back here at 45. So this is, in essence, the same thing that we did before on the test, but instead of using just a strand where I put cuts with, A and B, and I told you the size of the pieces, they could ask you to actually cut the DNA and estimate the size of the pieces. So that's just a little thing there. So the other thing, going back, this is just a crime scene one, and you'll notice that person 3 matches the crime scene. if it's a crime scene situation, the DNA would have to match exactly the crime scene. But if it's a paternity test, it's different. So I wanted to show you this. This is also part of your post-lab, and this will also be on your quiz. So here's how you do that. So this is really sloppy, I know, but this is mom, and this is the baby. Now, a baby is not going to be identical to the mom, because we're assuming this is regular sexual reproduction, mom and dad. You're going to have 50% approximately of your DNA in common with each of your parents. So here's what I'm going to do. I'm going to go to my baby, I'm going to look at their bands, and I see that this band matches mom, and this band matches mom. So those bands I can sort of ignore, because I know they came from mom, or at least they could have come from mom. That leaves me with these two other bands. These other bands, since they didn't come from mom, have to come from dad. So I have two suspects here, 1 and 2, and they want to know which one of these is dad. Well, both of these guys have this band, so that doesn't necessarily tell us anything. But only guy number 2 has this band, so he has to be our father. So again, I'll repeat, how do you do a paternity test DNA? Find the child. Take every band that came from mom, if mom's the one that you already know, and eliminate those because they came from the mom. Every other band the baby has to have come from the dad. So now you can go through all your possible dads, and you have to find a dad that has every single band that the baby didn't get from the mom. And that's going to be your guy. So that's how you do a paternity test electrophoresis, just so you know. And you will have to do something like that on the test. This is, again, just explaining the DNA, by the way, unlike our food coloring, DNA is not visible. You have to stain it. And so you would not be able to just look at the DNA right away. You'd actually have to either stain it with radioactive probes, which is what they use to make those X-ray looking ones. But there's some different DNA stands. What do we use it for? We can do crime scenes. We can identify bodies. We can do paternity testing. And we can do what's called genomics, comparative genomics. Genomics has to do with, for example, evolution. I could take the DNA samples from dinosaurs, compare them to several organisms that are alive today, and try to figure out, sort of build a family tree of what organism that dinosaur might be most closely related to. The idea is that the more close relative, evolutionarily speaking, we have, the more DNA we would have in common. The further back we diverged into different species, the less DNA we would have in common. And so this has been used to build all kinds of family trees. We're going to learn about this in our next chapter by comparing the DNA of different species. The last thing here, amplifying DNA, making copies of it. There's a technique called PCR, or polymerase chain reaction. It uses an enzyme from the hot springs. I'm just going to give you, it basically uses something called a thermal cycler. It heats and cools the DNA a bunch of times, and it makes lots of copies of it. You're not going to be tested on it, although they could mention it on the AP exam, so I'm just going to explain it real quick. So you heat the DNA up to almost boiling. Notice what happens, the DNA separates. In other words, the hydrogen bonds break. Then they cool it down, and they add primers. Remember, primers are used for replication. They cool it, the primers attach, and the primers now make a copy. So now we started with 1, we now have 2. Now they heat it again. So now I heat it, and notice my 2 DNAs separate, the hydrogen bonds break. I cool it, the primers attach, and now I've gone from 2 to 4. I heat it again, these 4 will separate, cool it, and 4 will become 8, 16, 32, 64. In 24 hours of thermal cycling, you have 1 million copies of the DNA. And this has really allowed us in crime scenes to take a small sample and make multiple copies of it. So whereas in, say, 1992, they might have been able to extract DNA and do a little bit with it, once they tested the sample from the crime scene, the sample was gone, and it could never be recovered. Now, if they get a little tiny piece of DNA at a crime scene, they can use this technique to make millions of copies of it. So whereas before they don't have enough to test, they can make enough to test and store it. So they can test some, store some, 10 years later they still have some on file. And so this has really, really improved our ability to use DNA technology, because we can make lots of copies of our DNA.